the gene expression. Just you understand the title, what is optimization? Otherwise called synonyms of optimization is called standardization. So why we make a cloned gene or cloning? Because we want some proteins for pharmaceutical applications, some protein for enzymes to production, or some fermentation process for some dairy industries, for some drug manufacturing, medicine, diagnostic, in space, our health, our routine lifestyle. So number of field are needed the biotechnology, biomolecules. Yes. For this purpose, we make the cloning to produce the particular proteins they wanted. They wanted for the corresponding applications. Okay. So situation is look like that. Then just we insert the foreign gene and identify and produce the number of copies of the clone. Yes, this is a non-complete process of cloning because we need specific requirements there. So specific applications are there. So we producing a specific protein for this corresponding purpose. Recombinant DNA technology effectively used in the biotechnology, especially applied biotechnology. Yes, biotechnology differ from recombinant DNA technology. Yes, biotechnology is simply called. I think nobody can knows for myself because I am go to somewhere in technology department. They are asking. Says anybody can define what is called biotechnology. Simply define. No, nobody can try to define full expression form of biotechnology, but simply say the techniques used in biotechnology. That is called biochemical techniques. Then why we are calling biotechnology? Technology used in biology. It is called biochemical techniques. Then we do biotechnology. So number of controversies are there, but anyway, no enter to that area. Just recombinant DNA technology used to applied in biotechnology for production of specific biomolecules. It's a new subject, not a new subject. Although it's a part of the biotechnology, for more attention than recently. Which subject is not new? The recombinant DNA technology is not a new. It's a part of the biotechnology used in the applied biotechnology, and it's get more attention because this is a backbone of technology we are producing, and the application oriented clones or organism or whatever it is. Biotechnology can be defined as the use of biological process in industry and technology. So, without recombinant DNA technology, the biotechnology cannot play the role. Yes, because we need a lot of biomolecules for industry purpose because they are making some lot of products, even from toothpaste to, even from flowers to, even from rocket engine, whatever it is, fertilizer, pesticides, costume, dressing, diagnostic field, antibody, medicine, drug, so number of vaccines, even cure of genetic disorders, even lifestyle, even automobile technology. So where whatever it is, biotechnology industries are produce molecules through by only recombinant DNA technology. For some time, using the archaeologists also. Archaeologists above 4,000 years ago, in the case of Neolithic period, late Neolithic period, they are using fermentation. So very oldest top field, biotechnology. And it's part of the recombinant technology. Very old field, but still we need some applications. Here I simply explain some examples. For example, Alexander Fleming, the very famous scientist, 1929, produced the penicillin. How we produce the penicillin? Through by the fermentation. But they don't know DNA or other compounds, cellular biochemical process are not involved to produce the penicillin. Just he's put a substrate and inoculum, finally get the penicillin. This in 1929. So the potent antimicrobial agent from fungi and bacteria, large scale production of antibiotics may be done through the microorganism. So this Alexander Fleming work behind the recombinant DNA technology, but he not known that time. Alexander Fleming not known that 1929 period. So nowadays require number of antibiotics. You know, uh, the Ranavir is a drug for coronavirus. It is not a suitable drug for coronavirus, but still antiviral drugs are needed. Even antibacterial needed. Staphylococcus aureus never killed by the canamycin. Is the ultimost antibiotics used. Nowadays, antibiotic resistant, multi-resistant bacteria are generated, or superbug bacteria are generated. So that continuously new antibiotics are required. But we can try to produce against the specific organism, the specific pathogen, even humans, plants, animals, or whatever. Next one is come to the point. So always recombinant DNA technology product is needed. Finally. A special vector for gene expression for foreign gene expression in E.coli, yes, special vector was needed. What kind of vectors we needed means we need expression vector, yes, a schedule vector is needed. Yes. Okay. Before we select the vector, we try to know that vector containing the optimization parameters are there. Either whether we inserted the gene into the vector, maybe it's expressed at the maximum expressed and produce the protein. Yes, we concern that point also when the time of we choosing the vector. So not easily choose the expression vector for the corresponding genes. Very toughest process. So we consider a lot of points, whether it's good, whether it's not, where the promoter is very weak or strong, where termination site is there, translation termination is there, the Shine-Dalgarno sequence is there, multiple cloning sites are there, either the origin of replication to make the copy number, either antibiotic selection is easily selected. So all of the factors we consider. Then why after that we can attain the maximum protein and the cloned gene, the expression product by attain. Otherwise we cannot attain the maximum production of cloned gene. product. That indicates its activation of clonal. Okay. Expressions depends on the total expression of the gene depends on the gene being surrounded the collection of signals that can be recognized by the bacterium. What surrounding by the signals means those signals came from upstream of the gene or regulatory element of the gene. Yes, generally genes divide in two parts. One is upstream, one is downstream. Downstream, all the part of the downstream maybe it's a coding sequences. Majority of the part is coding sequences. Remaining parts are UTR, untranslated segments, repeated sequences, intrans. So some of the other sequences are exist, but they are not regulated. They are not controlled genes. But the upstream segment of the gene having number of controlled sequences are there. Standard sequences are. That sequences are produce some signals. Some signal means even promoters. Promoter. Why we calling promoter? The promoter segment of DNA which promote the transcription of the corresponding downstream sequences. Yes. Operator. What is operator? An operator segment of DNA to operate the entire gene expression. That's why we call operator. So this is a specific segment of gene. Conserved sequences. Conserved sequences are there. Enhanced so number of sequences are there. So these are the surrounded collection of signals. These signals to control the rate of gene expression. So otherwise expression depends upon the signals. So these signals are give the instruction, provide the instruction for the transcription and translation apparatus of the cell. So three most important signals of E.coli genes are followed. So we are going to study three important signals. One is promoter. Number two is termination site. Third one is ribosome binding site. These important major signals which can drive the structural genes of the inserted or recombinant vector. So we choose the special vector for the gene expression. Okay, simply called three signals is called promoter, terminator, and RBS, otherwise called ribosome binding site. First we what is called, then we going to how we control the rate of the gene expression and how we optimize the gene expression. First what is called, promoter. Promoter means strong sequences present in the stream of the gene which can promote the transcription reaction. Yes. Without promoter sequence, the transcription never occurs. So when the transcription of the gene should started in E.coli should started by only presence of promoter. In E.coli, the promoter is recognized by the alpha subunit of the transcripting enzyme RNA polymers. You know very well prokaryotic transcription, prokaryotic translation, because Dori ma'am to tell more information. Yes, I am not to try to tell here. Alpha subunit of enzyme is it's called raw factor. It's called, simply I recall what is called raw factor. The raw factor which initiate the transcription reaction followed by RNA polymerase binding to the promoter sequence. Okay. Moreover, second sequence called terminator. This is a special sequence present in the end of the gene which terminate the transcription or translation, both. So this is a point of segment of gene, the end of the gene, which should stop the transcription. Terminator is usually nucleotide sequence that vasculars itself to form a stem loop structure. Yes. Where the stem loop structure is formed, whether this segment going to stop the translation reaction. Translation reaction. Yes. So the formation of stem loop like structure. So this considers mRNA. I think this is the end of the gene. This is the end of the gene. Considers this end of the gene. So when the segment of mRNA converted to the loop-like structure, the ribosome, the binding ribosome or polysomes, never move towards. So it's only stopped here. So never move. So after that, the transcribed proteins are released from some addition of termination factors. Yes. So terminator sequence is essential for optimization of protein expression. Yes. Third important sequence is called ribosome binding site. This sequence we already know very well. We already know that the sequence, other name of the sequence is called Shine-Dalgarno sequence. Shine-Dalgarno sequence. One important point I try to tell here. The ribosome consists of two parts, only small subunit and large subunit. Yes. So here mRNA is there. Wherever we see the picture, look like this. So this picture not look like this. First, mRNA is there. First, the small subunit goes to bind. Yes. So after binding on the small subunit of ribosome to the mRNA, then only the large subunit goes to bind and the structure look like this. So how the small subunit of ribosome goes to bind the specific mRNA means because presence of Shine-Dalgarno sequence in the mRNA. Okay. Shine-Dalgarno sequence is a conserved sequence. Standard sequence, conserved sequencer present in all mRNA, all eukaryotic mRNA. Same, whatever we isolate the mRNA up from prokaryotes, all mRNA contains same sequence of Shine-Dalgarno. No doubt of. Okay. So standard sequence are there. So same time, the small subunit first goes to bind the Shine-Dalgarno sequence. How means? The small RNA, the small ribosome contains 16S rRNA. 16S rRNA. This RNA plus 5S rRNA, other 60 number of proteins to made up of small subunit of ribosome. A small subunit of ribosome consists of RNA and proteins. Okay. A specific segment of 16S rRNA in the small subunit ribosome. This ribosome on the 16S rRNA, part of the 16S rRNA, which contains a complementary sequence of dead Shine-Dalgarno sequence. A complementary sequence of Shine-Dalgarno present in the 16S RNA. Then only this sequence go to complementary binds to the here. Suppose here A is there, here the complementary sequence are T. Yes, here is U, here is A, here is C, here is G. So the sequence of Shine-Dalgarno complementary to the specific segment of 16 rRNA in the small subunit of the ribosomes. So then only the ribosome goes to bind. So we ensure before we insert the gene. So the gene, the part of the gene contains must contains or should contains Shine-Dalgarno sequence. Then only the gene goes to express the maximum level optimization of the gene expression. These three parameters are very important for cloned gene expression. Optimization of cloned gene expression. Here he look like the structure. It's a promoter. So we concentrate the promoter, we concentrate on ribosome binding site, and we concentrate the terminators. These three are to control the rate of gene expression. Rate of gene expression. So remaining parts are there. Yes, remaining minor points are there, but they are not majorly control the gene expression. These are the three important parameters that control the major points which determine the rate of gene expression. So we try to learn one by one. Promoter, terminator sequence, and followed by ribosome binding. A typical promoter sequence or E.coli, the animal genes, is compared to the E.coli, the animal gene, and the promoter sequence of the particular gene. Because this is a conserved sequences. All the gene of eukaryotes and prokaryotes which contains promoter sequence. Okay, don't worry. So all promoter sequences look like the same structure, same DNA sequence. That's why we call it conserved sequences. Never we change. Never cannot change. Okay. First we consider as a data box. Data box. This is indicate -10 position. I think this is starting from age codon. Age codon is starting from 0. Here +1, +2, +3, +4 and goes on. Similarly here -1, -2, -3, -4 so on. Okay. So likewise the data box segment of gene exactly located -10th position of the upright upstream of the gene in E.coli. Similarly, in animals, from the start codon, the data box exactly located at -25%. But both species are both organisms which contains promoter sequence. Then only the genes going to effectively expressed. Second famous box is cat box. This is located between 0 to -35 position. Why we are calling box means it's a conserved sequence. Okay. That's very good. Similarly, here other kind of signals are there, but this is a minor factors to influence the beta gene expression. So typical promoter is needed, both expression of gene in E.coli or animal. Or animal gene cloned into E.coli. Whatever it is. Okay. The gene of higher organism are also surrounded by the expression of signals. Yes, don't worry. But their nucleotide sequence are not same. Nucleotide are not same. Exactly apart from the data box, cat box, and other sequences. In the E.coli. Yes. There are the similarities, but unlikely that on E.coli RNA polymerase would be able to attach the human promoter. Yes. Suppose we inserted eukaryotic gene into the prokaryote. we consider this promoter sequence. So why, how means, so we are inserted cDNA, maybe cDNA inserted a eukaryotic origin. This origin of eukaryotic gene into the expression prokaryotic and attained to optimization. How means that time we consider this promoter sequence. So E. coli polymerase is a machinery to control the gene expression. Don't worry. Yes. But here, human promoter is there because we have inserted the gene from eukaryotes. So it's contained maybe human promoter. The RNA polymerase having some ability because the RNA polymerase goes to bind, would be able to attach the human gene promoter also. Because both contains the same sequence, then only the eukaryotic, E. coli containing RNA polymerase goes to binds the human gene promoter. A foreign gene is inactive in E. coli simply because of bacterium does not recognize the expression signals. It's an important point. Whether the eukaryotic gene not expressed in the E. coli, that means the otherwise indicates the bacterium does not recognize the expression signals. This is the main important point. So, typical promoter is needed, whether what kind of genes you are going to do. Number 2, foreign gene is inactive in E. coli simply because of bacterium does not recognize it. How? How means because the problem would be inserted foreign gene into the vector in such set of E. coli expression signals. So kept your mind when the problem was arised, okay, because we contains human eukaryotic genes, the genes prokaryotic RNA polymerase goes to bind and transcribe. Okay, whether it's okay, but when we go attain the maximum expression or the foreign gene inactive in E. coli, that time we consider the cDNA or eukaryotic gene attached with the prokaryotic promoter. Yes, eukaryotic gene attached with the prokaryotic promoter. So the RNA polymerase goes to effectively bind and transcribe the gene. So don't the promoter not play the role. So for this case, so the time the foreign gene is active in E. coli and the RNA polymerase, the signals to recognize the expression and genes to be transcribed. When the eukaryotic gene contains prokaryotic promoter. Okay. Suppose the foreign gene is inactive in E. coli simply because bacterium does not recognize the expression signals. If this can be achieved, then the gene should be transcribed or translated. Cloning vector that provide a signal and can therefore be used in the production of recombinant proteins called expression vector. So expression vectors contains some relative segments promoter, Shine-Dalgarno, tag protein, and terminal tag. Finally, just we have multiple clones. Yes. So that multiple clones just inside the gene. So this inserted gene was controlled by the rate of expression was controlled by the promoter, the vector containing sequence. So then only that's why we are using expression vector. Here, very simple explanation is there. Okay, this is indicate promoter equal expression vector signals promoter ribosome binding site terminator. You should ensure before insert the generic expression. Promoters, very good promoters there. Either ribosome binding is there, yes. Terminator is there, yes. And also here multiple proline site is there. Whether we insert the T. Then only it's called expression vector. So we have inserted foreign gene, whether we insert it into host, so it can effectively expressed. Otherwise, 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 the expression is not attained at the maximum or optimization of technology expression not attained. So you consider these promoter, ribosome binding site, terminator here. So then only the genes effective expression. And also the origin of application to maintain the copy number also. So that time we produced multiple vector and the multiple vector recombinant vector. The vector produced the corresponding mRNA and the mRNA produced a corresponding protein. Then we can easily distinct the cells and isolate the protein. Whether the protein is intracellular, whether the protein is extracellular, automatically comes out in the fermentation medium. Then we recover the product. This is a use of an expression vector achieve expression in the foreign gene in E.coli. Yes. I think it's the last slide because other two factors we are going to study later. Now just I complete. The promoter is a crucial critical compound of an expression vector. The promoter divided into two parts. One is strong and weak. Strong promoter provides strong signals. Weak promoter produces weak signals. Yes. What kind of promoter we fused in the recombinant or expression vector? This is important to control the reduction expression. This promoter is an important compound of this expression vector because promoter controls the very fast stage of gene expression. Yes, and also due to the attachment of an RNA polymerase enzyme to the DNA. Yes. So by this, which determines the rate of which mRNA is synthesized and directly proportional to amount of recombinant protein will be produced. So that why we attain the maximum level. Yes. So without the strong promoter, we can never achieve this following consequence of recombinant steps. Promoter must be chosen very critical care. You choose very care. You take more care in for choosing the promoter sequence regarding choosing a vector. Lot of expression vector there. Which vector expression vector is good for this corresponding genes? You try to choose. Yes. Suppose if your host is a E. coli, you go to choose this sequences present in the promoter. This is a strong sequence of promoter sequence for E. coli clone. Then only you got optimization. I already told this is a strong and weak promoter. This strong promoter to create the platform binding of RNA polymerase very effectively. Weak promoter cannot easily attached to RNA polymerase. The attachment of RNA polymerase to DNA in the interaction is weak. So a small restriction or inhibitor concentration, if the inhibitors are suppose the protein product feedbackly inhibited means, so easily the RNA polymerase to stop of the binding to the corresponding promoter. So this promoter is called weak promoter. Yes. So only small amount of proteins are expressed. The only whose products are needed small amounts. Whether you are strong promoter, the large amount of products to be needed if you are using strong promoter. Okay. This is simply explained strong promoter or weak promoter. This is a gene. Okay, consider as a gene. This is a strong promoter, so weak promoter. So transcriptions begin. Strong promoter which transcribe the one more number of mRNAs at the same time. Similar, the weak promoter plays here the gene of transcription. After transcription, only one or few mRNAs are there because the number of mRNAs to determine the number of protein molecules, protein products are there. So this is the optimization, the maximization of gene expression. So this is only same proteins are, but the weak promoter contains to encode the low number of protein molecules. But if you are placing large number, if you get more recombinant protein. So thank you for joining. Thank you for watching. So other two factors we will be discussing later. Please unmute your